Hello, this is Harsha M, roll number 20E2509, and I'll be doing a poster presentation on bioprocessing and vaccine development. The history of vaccine begins with a fight against smallpox. Basically, a vaccine acts like a fire drill in the immune system as it triggers an immune response in the body. The body produces memory T lymphocytes and B lymphocytes that remembers how to fight the target disease in the future. Unlike natural infections, vaccines never cause illness, but they can cause other symptoms like fever, headache, body pain, etc. And it typically takes a few weeks for the body to produce the T cells and B cells after the vaccination. And depending on the type of antigen, vaccines are of different types, such as mRNA vaccine, conjugated vaccines, live attenuated vaccines, inactivated vaccines, etc. Even though antigen types vary among different vaccines, the vaccine manufacturing process in general is similar for all types. A typical vaccine manufacturing process includes upstream, downstream, and formulation steps. An antigen is generated in the upstream process. It can be the pathogen itself, or it can be an antigen generated from a recombinant protein. This antigen is responsible for inducing an immune response. The antigen is then separated from impurities in the downstream process, the second process. The antigen is released from the substrate if necessary and isolated from the bulk environment depending on the characteristics of the antigen and the impurity. Purification technologies like chromatography, ultrafiltration, precipitation, enzyme digest may be employed. All components that constitute the final vaccine are combined in the formulation process, the final process. The formulation is designed to maximize the stability of the vaccine. The formulated vaccine may include an adjuvant to enhance the immune response, stabilizers to prolong the shelf life, and preservatives that ensure that multi-dose vials can be delivered. This was a basic overview on manufacturing vaccines. Now moving on to processing and production of viral vaccines, which is the main topic of this poster. Virus is basically a microscopic infectious agent that can reproduce only inside a host cell. Viruses are generally attenuated via passage, growing several times in unrelated or foreign hosts such as tissue culture, embryonated eggs, or live animals. Likely, one of these will possess a mutation that enables the virus to infect the new host. However, this mutant normally has a lower virulence than the virus that was in the original host, the first host. The genetic information for interacting with the host does not change, enabling it to infect the host, but it causes less damage and so acts as a vaccine. The live attenuated viral vaccine manufacturing follows a complex multi-step process. The manufacturing process for each viral vaccine is different, and the general outline of the process is summarized in this poster. So, the initial steps involve the thawing of cells and cell expansion in a bioreactor. Then there is virus production, where viruses are propagated in cell culture. There are several types of cells used for growing viruses for vaccine applications, such as human diploid and Vero cells. Vero cells, which are basically derived from African green monkey kidney cells, are most commonly used for viral vaccine manufacturing. Most cell cultures for viral vaccine applications are grown in low oxygen tension in the presence of approximately 5% of carbon dioxide. Virus inoculation is done aseptically to cell culture that has grown for 5 to 7 days. Virus harvesting is done after 24 to 72 hours of virus inoculation. Depending on the virus type, they either bud out of cells or lyse the cells and emerge in the extracellular culture fluid. In some cases, the cells need to be lysed by the addition of detergents or surfactants to release the viruses from the cells. Next step is clarification. Clarification removes the cells or cell debris and harvests viruses. Zonal centrifugation is commonly used for primary clarification. It is basically a centrifugation technique employed to effectively separate particles of different sizes. Some manufacturers also use tangential flow filtration under low shear conditions or normal flow filtration, in most cases depth filtration, for clarification of viral vaccines. Some attenuated live viruses sometimes tend to bind to cell surfaces or get trapped in lysed cell debris. This leads to their removal during clarification, which also results in poor virus recovery. Next step is the nuclease treatment. Carryover nucleic acid, that is nucleic acid released from lysed cells, is a key contaminant in viral vaccine process. Viruses propagated in human diploid cells or non-human cells pose a greater risk of nucleic acid carryover. Benzonase nuclease is an endonuclease that is commonly used to degrade the nucleic acids such as RNA and DNA of the host cells. After benzonase endonuclease treatments, the harvest is filtered to remove benzonase endonuclease and degraded nucleic acid components. This treatment is sufficient to bring most attenuated viral vaccines to the desired level of purity during the concentration and diafiltration step. However, chromatography is normally required to bring new generation of viral vaccines such as dengue virus to the desired level of purity. As viruses are negatively charged, anion exchange chromatography works well in bind and elute mode. Post chromatography, the eluted virus is concentrated by using tangential flow filtration devices. Final virus vaccine bulk is basically comparable to that of water. During final filter, vaccine is filter sterilized. Many of the attenuated viral vaccines are finally formulated with different strains of the virus. Most of the live attenuated viral vaccines do not need any adjuvants because they are naturally potent immunogens. They can produce immune response by themselves. Most of these vaccines are lyophilized, that is freeze dried. The final formulation of attenuated viral vaccines contains a small amount of antibiotics, excipient, stabilizer and buffering agents. Excipients are active substances that serves as a medium for a drug or other active substance. Most of the vaccines are administered subcutaneously except for rotavirus and poliovirus vaccines, which are isotonic solutions that are administered orally. So this was a basic overview of vaccine bioprocessing and its development. Thank you.